reaction takes, particles must overcome activation energy. Activation energy is the energy barrier which must be overcome before the reaction will take place. So like for example now, look at this is our activation energy. So this AB must cross this activation energy before A plus B produce. So what this reaction does, or what this enzyme usually does, they reduce this activation energy. That is how they speed the rate of the biological reaction. And as I said, each enzyme is responsible for catalyzing one type of reaction. So if we have 1,300 enzymes, it means that there is about 1,300 reactions. And in one time, or in 1 minute, or in a second, millions of complex reactions are taking place in a human cells. Because in human cell, we have various reactions that range. We have various range of reactions that are taking place simultaneously in a human cells. And this reaction includes balance in endocrine system. What is balance in endocrine system? Remember that when we said endocrine system, it means hormones. And hormones are chemical messengers that brings about coordination. So some hormones are responsible for controlling sexual characters, and some hormones are responsible for controlling growth, while some hormones are responsible for controlling our intelligence. So as some of these hormones are doing these particular functions, other enzymes at the same time, or another hormones at the same time are also carrying out another reaction in different part of the cell. That is why, as I said, in just very few minutes, you will surprise that millions of reactions might take place in a body. So these reactions that are taking place in our body include fuel utilization. Fuel utilization include glycolysis. Remember that we need energy. You that or generally nurse are working in the hospital where some people where you have some patient as a result of lack of their body to produce enough energy. So you know that if you are not taking food or if you are not taking your diet, you may have some disease develop in your system. So if your body is not properly utilizing fuels, there is likelihood that you may actually come up with some disease. Immunity also, in immunity. Immunity is like a kind of soldiers, it's like an army that we have in our body that always try to protect us from disease pathogens, disease agents. And we have digestion. Digestion is also a type of reactions, or is like a system that involve combustion of a large molecule of food into smaller ones. So all these are reactions. And we have homeostasis. Homeostasis is like the control of an internal, organ of a living organism. So if a body system is not properly controlled, then you will develop a disease. Like for example, we have what you call, let's take example with diabetes. The normal range of normal blood glucose in the blood is within 4 to 5 milligram per deciliter. So if the blood glucose is above that 5 milligram per deciliter, we call the condition hyperglycemia. Because when there is this increase, the body must adjust so that to control and maintain that changes of the blood glucose level within the stable state, which is within 4 to 5 milligram per deciliter. So that is why whenever there is an imbalance, there is a change. Look at it. We have a homeostasis here. So whenever there is an imbalance, or there is a change, a receptor will detect the change. And then that change will now send to a control center like the brain. Once the control center receive a message from the brain, then what will happen? So, sorry, once the brain, which is the control center, accept or get the information from the receptor that there is a change, then the brain, which is the control center, will now send a message to the effector that there is a particular change. And then that effector will now act on that change and respond to it. And then you maintain balance. Like, for example, when you are infected with some particular microorganisms, like, for example, let's say you are infected with coronavirus. So when you are infected with coronavirus, our normal body temperature is 37 degrees centigrade. Our normal body temperature is 37 degrees centigrade. So when you are infected with coronavirus, because most of these virus and the other and these pathogens, they survive based out at our normal body temperature. Then once we are infected, then a message is now sent to our hypothalamus that there is a problem. Remember, hypothalamus is responsible for controlling body temperature because of the presence of the thermostat. The thermostat is actually what does the function in the hypothalamus. And then the thermostat will now unset the temperature and increase it to around 30 to 40 degrees centigrade. So once our body temperature is increased, then what will happen? Those microorganisms or viruses will not actually survive that temperature. Then they will die. And once they die, your body will now go back to its normal body temperature. So in summary, from this point, what I want all of us to understand is that in this biological system, we have number of reactions. And it's this reaction that make us to survive. And if there is any problem in one particular reaction, there must be a disease if there is a problem in a particular reaction. Like, for example, let's take example imbalance endocrine system. If you are not producing enough growth hormones, there is a problem. If your immunity is not functioning enough, there is also a disease. So that is what I want us to understand in this slide. There are a number and millions of reactions that are taking place in our body in order to ensure that normal life process, there is functional system. Okay, then now we are moving to the next slide. Also, this is also another summary of what we did last week. All these things, I discussed them last week. What I said is actually when you have sperm cell and egg cell, and when actually a fertilization took place, we have a zygote. And in this zygote, we have a DNA. And in this DNA, a zygote is an undifferentiated cell. It's a cell that it's not specialized. It's a cell that is not specialized. So how this cell specialized is because in this zygote, there is a DNA, and in this DNA, there are genes. And these genes are responsible for producing different proteins. And some of these proteins have structural function. So all proteins that have structural functions, they fall under physiology. And some of these proteins have some of these proteins have functional ability or functional activities. Like, for example, we have proteins that allow us to see, like opsin and crystalline. Opsin and crystalline are the proteins that are produced from the DNA that allow us to talk. So we call them functional protein. Here is supposed to be functional. It's not structural. It's not structural. It's functional. So some proteins that are produced from this DNA have functional activities. Like, as I said, we have FOXP2 protein, which produced from FOXP2 genes. It's a protein that make us, we human, to talk. So that is why we talk as a human. So it falls under physiology. And we have some proteins that are produced from this DNA that they have structural function. That is why they fall under anatomy. So remember, the shape of our red blood cell is biconcave in shape. And what makes it biconcave is because of the proteins that are produced from the DNA. And the name of the proteins that are found in the red blood cell that make it to appear as biconcave is hemoglobin. And we also have some proteins that make our nose, or the shape of our nose, the shape of our eyes, the shape of our ear. We have some proteins that are responsible for controlling that particular structure of all these organs. So all these proteins are produced from DNA. They are produced from DNA. So that is why physiology and anatomy are dealing with proteins, and these proteins are under biochemistry. So now, as I'm saying, if you have a zygote is not specialized cell. It's not a specialized cell. After women conceived, or when women are pregnant, remember there is an embryo under embryogenesis. This zygote cell is the cell. When you have a zygote cell, this zygote cell will undergo mitosis to produce 2, and from 2 you have 6. Sorry, you have 2, from 2 you have 4, from 4 you have 16, and from 16 you have 32, and from 32 you have 64. So they will continue dividing themselves. So when zygote cell divide itself to produce, or to divide into 16, into 16 cells. So a 16 zygote cell that produce from one single cell are called morula. So we have morula and blastula before embryo. So embryo is like a sum of a specialized cell. It's like a sum of specialized cells from zygote. So, but how? Because it's from those zygote cells that are not specialized that they will be instructed to produce a proteins that are responsible for producing eyes. And from that, the same zygote cell that do not differentiate themselves, that they are unspecialized, and some of those cells will also be instructed to produce what we call brain cells. Some will specialize to produce kidney cells, some liver cells, some eyes, some mouth, some legs. So all these proteins are specialized as a result of the productions or differentiation of gene expressions of those cells. Our brain cells, or the proteins that are produced in brain cells, are not the same with the proteins that are produced in the pancreas. Remember, in pancreas, we have insulin and glucagon. They are proteins that are produced from the DNA, and that is what makes them to specialize as a pancreatic cells. In the brain, we have neurotransmitters and some proteins that make the brain use in controlling the intelligence. So this is how cells are specialized. So unspecialized cells are called stem cells. And these stem cells have the potential to produce all different cell types. For example, is these stem cells that are produced at the zygotic level that can produce blastocysts, cardiac cells, erythrocyte, fat cells, red blood cells, chondrocyte, and epithelial cells. So this is for this slide. So this is just a summary of what we discussed last time. Then what are the breakthrough in biochemistry? The breakthrough in biochemistry include one, the discovery of DNA, the discovery of DNA. A DNA discovery was made in the year 1869 by a German scientist. A scientist from a Germany called Friedrich Mister. Friedrich Mister is a German scientist that discovered DNA. And let me tell you, ladies and gentlemen, since the discovery of the DNA, a lot of things have changed in science. A lot of things have changed in science, including modern medicine. A lot of things have changed. And then after the this Frederick Mister was able to discover DNA, and then the next scientist is Levin, and is a scientist that was able to demonstrate the molecular structure of DNA, which is defined by a lattice. So the DNA is made up of some lattice from the DNA, or these lattice, or these four lattice are the building blocks of DNA. We call them nucleotides. Adenine, guanine, cytosine, and thymine, they are the nucleotide that was discovered by Levin in 1919. And then the next thing is another scientist called Colin MacLeod and Mackie. He is the scientist that was able to find that DNA carries genetic information. It means that the inheritance that we have from our parent is as a result of DNA. If you are short, it's because your parent have the DNA that code for short character. If you are intelligent because your parents are intelligent, and the intelligence is there in the DNA of your parent. And ladies and gentlemen, let me tell you, intelligence is usually, or 80% of intelligence is coming from mother. Due to breastfeeding, XS chromosome, and then the mitochondrial DNA. So that is why 80% of the intelligence is coming from DNA. So don't think that you are intelligent and you marry a dull woman. So there is a likelihood that 80% to 90% of your child will not be intelligent. So intelligence is coming from DNA, and 80% of it is from the mother. And then the next one is another scientist called Erwin Chagall. Erwin Chagall is the scientist that was able to demonstrate the base pairing, where adenine pair with thymine, and then cytosine pair with guanine. So cytosine pair with guanine. So with time, we'll look at all this. And then the next thing is another discovery in 1953 by a scientist called, there are two scientists, Watson and Crick. So James Watson and Francis Crick are the scientists that was able to demonstrate the double helix structure nature of DNA. So DNA is double stranded with helical structure. So Watson and Crick are the scientists that was able to demonstrate that the structural nature of DNA. So that is this. And then the next thing that we are going to look at is the Human Genome Project. So the DNA in the human DNA is very large, very, very large. Is very large. If you can take the whole of DNA from human, the whole of DNA in human cells, and you stretch those DNA, they can reach from the surface of the earth up to sky. But because of the thin nature, the DNA is very thin. It's very, very thin. So that is why it's tightly wrapped and twisted and fits into the nucleus. So the DNA is very huge. It's very huge. It's lengthy. But it's just because of it is that thin nature, that is why it can easily manipulated and twisted and fits into the nucleus. And where do we have this DNA? Remember, in a cell or in a human being, we have a cell, and in a cell we have nucleus, and in that nucleus we have what you call chromosomes, and in the chromosomes we have a DNA, and this DNA is tightly wrapped with a protein in that chromosomes, and the name of the protein that tightly bound with the DNA is called histone protein. So DNA is negative charge. Ladies and gentlemen, put this on your note. DNA is negative charge, while histone protein is a positive charge. So that is why there is a strong interaction between positive and negative. So DNA is negative and histone protein is positive. So that is why they can interact. So histone protein is tightly wrapped this DNA, encapsulate it and capture it into itself. And then what is the Human Genome Project? So a human project is the entire set, or when we say genome itself, a genome is the sum of the genetic material that are found in a human cell. So I'm starting with the genome. A genome is a summation, is a sum of all the DNA, is the entire set, of DNA that are found in a particular living cells. And the genome we have in human being is different from the genome that are found in cats, rats, and other organisms. And this is why we are different. So we are different from all other species of organisms because of this nature of genome. So ladies and gentlemen, with this, you will understand that Human Genome Project is very, very important because in Human Genome Project, we are just trying to understand how this DNA look like, how those letters that I mentioned, which is adenine, guanine, cytosine, and thymine, how are they arranged? So now, ladies and gentlemen, I also want you to put this on your record. Where do we find DNA in a cell? So from here, you can clearly see that in a cell here, we have DNA in a nucleus. So apart from nucleus, we also have some other part of a cell where we have DNA. So we also have DNA in mitochondria, and we also have DNA in chloroplasts. So DNA is found in three parts of a cell. It's found in a nucleus, it's found in mitochondria, and also in chloroplast in plants. So this DNA or deoxyribonucleic acid, it carries the complete genetics information responsible for the development and functioning of an organism. So ladies and gentlemen, let me tell you something here very important. If I can get a DNA of one of you, let's say I take the DNA from Fatima Shitu. If I can take a DNA from Fatima Shitu, I can produce the same Fatima, the same color, the same height, the same everything, the way she will talk, because DNA is the language of life. So Fatima Shitu, you are the way you are because of your own DNA. If you are beautiful, it's because of your own DNA. If you are not beautiful, it's because of your DNA. If you are short, it's because of your DNA. If you are intelligent, it's because of your DNA. So DNA is very, very important. It's a language of life. You are the way you are because of your own DNA. So if I want to produce Godia Bala, or if I want to produce Victoria, I can just get her own DNA. Or if I want to get, if I want to produce, or if I, with a DNA, I can produce Maimuna Sheikh Musa, I can produce her with her own DNA just to get her own hair. From her own hair, I can isolate her DNA and construct the same Maimuna Sheikh Musa. And I can, from the DNA of Mariam Yusha'u, I can produce her. So that is cloning. So all this is possible in science. So that is why DNA is everything, is everything. People develop genetic disease is because of the problem from their own DNA. Secular is problem from DNA. So now all this information are being obtained or are being known after genome sequencing, after the sequencing of human genome. So in a human genome project is just a project that tries to understand how the entire, how the entire letters, how the entire letters, adenine, cytosine, guanine, and thymine are being arranged. So genome sequencing is a process of determining the complete DNA sequence of an organism's genome. So
It
genome
involves
sequencing is
identifying
the process of determining the arrangement of these letters. So it involves in identifying the order of base pairs, the order of these base pairs. How are they arranged? So now, ladies and gentlemen, when this Human Genome Project was conducted, the Human Genome Project was started in the year 1990 and completed in the year April 2003. So it means that it took science it took scientists 13 years to have a complete human genome project. And let me tell you, ladies and gentlemen, as I told you, DNA, DNA is very long. It's very long. It's huge. So because of that huge nature of the DNA, scientists are not all, even scientists from one particular country was not be able to, to carry out human genome project. So the DNA is being dissected. The human DNA is being dissected, being cut into 6 fragments. One fragment is taken to France, another fragment is taken to Germany, another fragment is taken to China, another fragment was taken to Japan, another fragment was taken to UK, another fragment was also taken to China. So this is how the project is being sent to different parts of the world in order to carry out the human genome sequencing. So Human Genome Project is an international program that leads that leads to the decoding, unveiling, knowing the entire human genome. Human Genome Project, HGP, gave the ability for the first time to reach nature's complete genetic blueprint of building a human being. That
comparar com o mutum ya
is by knowing the DNA of particular individual, you can produce that particular or that complete human being. Please, who is that making noise? Can we? Unmute yourself. Okay. Then now, what are the applications of human genome projects? So since after the sequencing of human genome, scientists was able to successfully diagnose disease and treat the disease. So disease diagnosis and treatment identify the genetic mutation and variation that contributes to disease condition. So since then, nearly 1,000 diseases were able to identify due to a single gene malfunctioning. So after the human genome project, scientists was able to identify and treat a lot of diseases, and the etiology, that is the causes of nearly 1,000 different diseases, have been known. And most of those diseases are not due to the presence or due to single gene malfunction. Like, for example, people are developing sickle cell disease due to a single mutation or a mutation in just one single gene. So what is that? And it reads the code of viruses. In 2015, of scientists from MIT and Harvard University sequenced samples of a from infected African patients to show how genetic data of viruses could reveal hidden pathways of transmission. So you see, since the Human Genome Project, scientists was able to read and code, so they was able to read and sequence the complete genomes of the virus, which make them to unravel or to get a lot of information. Okay, ladies and gentlemen, we are going to rejoin this meeting now because my upgraded, so it's just for 40 minutes.